Hello everyone, today I'm going to introduce our work on investigating magnetic susceptibility of human knee joint at 7 Tesla. QSM has been mostly applied to the brain studies, and recently it has been applied outside of the brain to the body, such as the liver, kidney, heart, and bone. By rotating the tissue at different orientations relative to the visual field, our group has shown the susceptibility anisotropy of the white matter is mainly from myelin. We also show the susceptibility anisotropy of tubes in the kidney. So how about the susceptibility anisotropy of collagen fibers in the cartilage? The purpose of this study is to quantify the magnetic susceptibility of knee joints, as well as to measure the collagen matrix and to describe the underlying 3D collagen fiber networks. The data was acquired on a 7 Tesla MR scanner using a 28-channel knee coil with a 3D flash sequence with the scan parameters as below. The TR is 35 milliseconds and the first T is around 2 milliseconds with echo spacing 2.2 milliseconds. The spatial resolution is 0.4 by 0.4 millimeter with slice thickness 1.6 and the Guarpa factor is equal to 3. So the scan was repeated 2 more times with first T increased by 0.3 milliseconds between acquisitions, giving a total of 3 scans. Then the 3 scans were combined for the later water-fat separation. Here is the processing pipeline to estimate the susceptibility map of the knee joints. First, the complex multi-echo images are used to estimate the visual field map, water, and fat. A knee mask is derived from the magnitude image. Then the local field is obtained by removing the background field. Lastly, the QSM was reconstructed by using star QSM method. Here is an example of GRE image in a healthy volunteer. A is the magnitude, B and C show the QSM image with and without chemical shift removal. With water-fat separation, you can see much clearer susceptibility contrast between different tissues, such as between muscle and cartilage. E is the estimated water, and F is the reconstructed susceptibility map, corresponding to the area outlined in the white box on the magnitude image. Both water and QSM maps identify a portion of the nerve just behind the knee. QSM map also shows the structure of the artery. Red arrows indicate the artery wall reviewed by QSM. We show that the cartilage exhibit clear layer structure with different magnetic susceptibility values at different depths. The susceptibility increases from deep zone to the superficial zone, and the likely source is the visual orientation dependency of the magnetic susceptibility of the collagen fibril. Since the collagen has a very well organized layer structure, specifically in the deep zone, most of the collagen fibers are perpendicular to the surface, while in the superficial zone, most of the collagen are parallel to the cartilage surface, while in between the collagen are relatively randomly distributed. We also show that for the diseased cartilage, the susceptibility contrast changes quite a lot compared to the healthy knee. To further confirm the observed magnetic susceptibility anisotropy, we also conducted the ex vivo experiments using a piece of pig cartilage that rotated at different orientations. As we can see here, the susceptibility value at different cartilage depth change a lot at different orientations. On the left is the apparent magnetic susceptibility maps that occur at 15 orientations with respect to the visual field. As you can see, the susceptibility map exhibit local contrast specific to different layers, which shows very strong dependency on collagen fiber orientations within the cartilage. Quantitatively, plots of magnetic susceptibility measurements as a function of the angle between the normal of the cartilage surface and the visual field in different layers are shown on the right. Specifically, negative correlation was found in the superficial zone, and positive correlation was observed in the deep zone. There is no visual orientation dependency in the middle zone since the relative random distribute collagen fibers and the magnetic anisotropy that cancel each other. To validate the observed susceptibility anisotropy, we simulate the microstructure of the collagen using a 10+4 model. A and B on the left are the electron microscope image of fibrils of the human tendon. A single collagen fibril contains 14 microfibrils, with 4 in the center and 10 in the outer space, as shown in Figure C. Each white circle represents a microfibrils that contain 5 right-handed triple helix, as shown in the Figure D. And this right-handed triple helix is constructed by 3 left-handed peptides. The basic units of the peptides in the collagen are the planar peptide ground as shown in Figure F. Each can be assigned with a second-order tensor with 45-degree angle respect to the long axis, and the tensor was rotated according to its position in the right-handed triple helix. We rotate collagen fibril at different orientations relative to the visual field at different echo times. A is the collagen mask shows the 10+4 model, and B is a calculated apparent frequency with collagen fiber perpendicular to the visual field. We draw 2 ROIs. One is inside the collagen, we call it intrafibril ROI, and another is a little bit far away from the collagen, called actual fiber ROI. The magnitude and the mean apparent frequency within each ROI at different orientations are plotted over echo time. We also plot the magnitude and the mean apparent frequency in the entire simulated volume, as shown on the right. So the results showed a very strong susceptibility anisotropy of the collagen. Moreover, the anisotropy is much larger in the extrafiber space than intrafiber space. The results also showed a T-dependent frequency shift and susceptibility. This T-dependent frequency shift could be contribute to the different T2' components in different locations. As we can see here, intrafiber space has a shorter T2' and signal decay faster compared to the actual fiber space, leaving the actual field perturbation to dominant the measured frequency. As a result, the complex average of all spins within the entire simulated volume yield a T-dependent susceptibility. Here we can see the 3D collagen network in the cartilage from different views. The collagen fibers are parallel to the cartilage surface, and the blue fibers are mostly perpendicular to the cartilage surface, while in between, since the midzone has relative random distributed collagen fibers, we can see the tractography fields in the midzone. To our knowledge, this is the highest resolution tractography that shows the 3D network of the collagen fibers in the cartilage that can be achieved using MR until now. To conclude, the susceptibility takes advantage of collagen fibers and associated proteins in the cartilage. The cartilage is magnetically anisotropic. The likely source is the triple helix peptide bond. And we build a model and confirm what we observed using the 10+4 model. And in the future, we think this may be early detected by magnetic susceptibility. Thanks for your attention.